Hello, students. In this lecture, we look into the gene silencing. So, RNA interference is one of the important mechanism involved in gene silencing. So, we'll look into the mechanism of RNA interference in this lecture. So, gene silencing, we are aware that when you want to inhibit the expression of a particular gene, that is, when you want to stop the synthesis of a particular protein being synthesized from a gene, you go for the inhibition of this gene. So, RNA interference is one of the important mechanism used in vitro for gene silencing. So, this RNA interference is a mechanism which is found in vivo. So it is an evolutionarily conserved mechanism that causes sequence-specific gene silencing and the trigger molecule being double-stranded RNA. So it is a mechanism which is found in vivo. And why it is being evolved in vivo is to mediate resistance to both endogenous and exogenous pathogenic nucleic acids and to regulate the expression of protein coding genes. So the function of this RNA interference in vivo is to regulate the gene expression and to mediate resistance to endogenous and exogenous pathogenic nucleic acid, especially double-stranded RNA. Now, these are the two uses of RNA interference in vivo, and in vitro, we are successfully using this as a tool in functional genomics to manipulate the expression of a desired gene. And this RNAi mechanism has been discovered in various organisms. So it is being discovered in various organisms, and we can find that the major trigger molecule being double-stranded RNA or an exogenous molecule, a transgene. So this triggers the silencing phenomenon, which is known by different names. So this RNA-mediated silencing is known as co-suppression or post-transcriptional gene silencing in plants, quelling in fungi, and RNA interference in the animal kingdom. So the most important feature of RNAi can be summarized as the double-stranded nature of the silencing molecule, the presence of catalytic and amplification process, which is indicated by requirement of only few molecules. So the whole silencing phenomenon requires only few molecules to silence the whole transcript, the mRNA. The stage-specific silencing, which is heritable and systemic too, and this silencing at post-transcriptional level is can be influenced, okay, by altering the chromatin structure, also the chromatin remodeling. So it is out of scope of this lecture, but yet the studies in yeast and tetrahymena indicate that this RNA interference can also influence the transcription by altering the chromatin structure. And this RNA interference is extremely specific mechanism. The important features of RNA silencing, though it is PTGS in plants, quelling in fungi, RNAi in animals, they share a critical common component and features in the process. So the inducer is essentially a double-stranded RNA molecule. The target RNA, that is your mRNA, is degraded in a homology-dependent fashion. And the degradative machinery requires a set of proteins which are similar in structure and function across the most organisms. So when you come down to the components of gene silencing, the dicer is a major molecule, key important molecule, which is needed for the RNA interference, the dicer. It is the RNase 3 nuclease and is involved in the production of small interfering RNA from long double-stranded RNA. As you can evidence from this illustration, so there's a double-stranded RNA is bound, and what happens? They are getting converted into siRNA. This dicer family proteins are ATP dependent nuclease, that is, they require ATP for their function. And this RNase 3 enzyme, the dicer, acts as a dimer, two molecules. And the dicer homologs will exist in many organisms, including C. elegans, Drosophila, yeast, and humans. The importance of this dicer molecule is learned by the loss of this molecule in vitro. So there was a loss of silencing when there was loss of dicer. This dicer is a RNase3 family, as already indicated, and it has four distinct domains. So the domains being an amino terminal helicase, a dual RNase3 molecule which is found at the carboxy terminal, and a double-stranded RNA binding domain. And the key domain for interaction is your PAS domain. So this is a 110 to 130 amino acid domain which contains proteins like ergo, PV, aragonate, PV, and zwili, and these are thought to be important for protein-protein interaction. So all these are the major components of your dicer molecule. So the dicer converts the double-stranded RNA into the small interfering RNA, which are the effector molecules of this RNA interference. So these are essentially 25, approximately 25 nucleotides, which are of both sense and antisense polarity. The siRNA, as they are formed and accumulated as double-stranded RNA molecules of defined chemical structures. The key features of this siRNA, you find that you have a 5' phosphate group end, you have a 3' OH end, and the features are that another essential feature is that you have a 2 to 3 nucleotide overhangs at the 3' end, which is a key feature of the siRNA. The effector molecules are the siRNA, and the catalytic component is your dicer. Now, both of these together are loaded to form the RNA-induced silencing complex, that is RISC. So this RISC is a multi-component, right? Multi-component complex, which is of about 500 kilodaltons, and it contains the member of aragonate family, RNA binding domains, a DFMR protein, RNA helicase, and two ribosomal proteins. So this sequence specific nuclease, that is your siRNA, it is loaded as such into this complex, and it guides into the target specific mRNA. So the sequence recognition, that is the complementarity, is what the key feature over here. The siRNA identifies the target mRNA through its sequence complementarity and binds to it. And the cleavage of this cognate mRNA is placed near the middle of the region which is bound by the siRNA. So the siRNA identifies and binds. So it guides the complex to the mRNA which has to be degraded. It binds, and then the cleavage is effected. The schematic illustration of the actual mechanism of the RNA interference. So let us go through this in by step. So the active molecule which affect the RNA interference mediated gene silencing are obviously the small interfering RNA, siRNA. The first step is the production of this siRNA by the dicer molecule, which cleaves the long double-stranded DNA into approximately 25 nucleotide small pieces of siRNA, which occurs in the cytoplasm. And these siRNA are now incorporated into the multi-protein silencing complex called the RISC, RNA-induced silencing complex. So this siRNA is loaded into the RISC. Now, once the siRNA duplex is loaded, what happens is there is a conformational change. Since we have the helicase molecules there in the RISC complex, we find that there is helical conformation being changed, the active conformation being generated. Once there is conformational change, the recognition of the mRNA occurs. So it is the mRNA to be degraded now the recognition is mediated by the antisense RNA, that is the siRNA, which acts as a guide RNA strand for the RISC complex, and this mediates a endonucleolytic cleavage of the homologous RNA, that is your target mRNA. The target mRNA is now cleaved in the center of the region, And this is by the sequence complementarity, as we have already seen. So it is actually 10 to 12 nucleotide from the 5' end of the siRNA. Now, the final step in this process is the amplification of the siRNA molecule itself. So the siRNA, what happens is there is a priming by the RNA-dependent RNA polymerase on the target mRNA by existing siRNA. So you have the generation of secondary RNAs, siRNA. Now these are able to induce a secondary RNA interference, which is also known as transitive RNA interference. So the mechanism essentially is so simple. The dicer molecule converts double-stranded RNA, which is the trigger molecule usually, and these SIR RNAs are found in the cytoplasm. Fine. So dicer molecules slice the mRNA, the double-stranded RNA, into smaller siRNA, and now these are loaded onto the RISC. So this RISC complex then undergoes a conformational change upon binding of the siRNA, and this is now led to the or being guided to the mRNA, which has to be degraded. The sequence complementarity of the siRNA and the mRNA, target mRNA, is a key factor over here. So upon binding, there is endonucleolytic cleavage of this target mRNA. So you can also have the secondary amplification of this siRNA molecule itself. So these are the key features of the RNA interference in short. Now, where does this find application? The major application, it is actually a important tool of reverse genetics. So it is useful for analyzing unknown gene in sequenced genomes, identification of the gene function. Obviously, when you silence the gene expression, you can understand, you can find out the missing function, and this can be attributed to the gene which has been silenced. So the identification of the gene function through RNA interference. And it finds a major role in gene therapy, where you can downregulate certain genes or mutated alleles, which are the key factors for the disease, actual disease. The gene being the reason for the inherited disorder can be downregulated or it can be silenced. So in cancer treatment too, you find the application of this RNA interference to knock out the genes required for cell proliferation, the cancer cell proliferation, or you can knock out the genes which are encoding the key structural proteins in cancerous cells. And the other major function of this RNA interference is in the field of agriculture for production of plants with desired traits. For example, this RNA interference can be utilized to delay fruit ripening, wherein you can knock out or the gene, silence the gene, which is essential for ripening of the fruit. So, which will extend the keeping quality of the transgenic product. So, these are the various applications. Hope you had the understanding on the basic concept of RNA interference and its application. Thank you.